All right, welcome. My name is Jonathan Holtquist, and this is Vancouver Aquarium Marine Science Center. Canada is an Arctic nation, yet only a small percentage of Canadians actually live there. So how do people living in the southern parts of Canada, or anywhere else for that matter, learn about the unique and fascinating marine animals that inhabit this enigmatic environment? Let me present to you the online Arctic Marine Life course. The course is supported by the Science in Canada's North Café Scientifique series, and we'd like to acknowledge the support of the W. Garfield Weston Foundation and the Canadian Association of Science Centers for their support. Our partners include the Alliance of Natural History Museums of Canada, Canada's Accredited Zoos and Aquariums, Telespark in Calgary, the Canadian Museum of Nature in Ottawa, and Discovery Centre in Halifax. You can keep track of the dates and locations of presentations on our website at www.vanaqua.org/Arctic Marine Life course. If you miss a presentation, you can always watch it online after it's been recorded. And if you have any questions about the course, you can contact me at jonathan.holtquist@vanaqua.org. Our speaker is Dr. Marie Noel. She is the manager for the Ocean Pollution Research Program at the Vancouver Aquarium Marine Science Center. She studied marine mammal toxicology at the University of Victoria and documented the effects of contaminants in the health of beluga whales in the Beaufort Sea. Dr. Marie Noel.
Good evening, everyone. Thanks for coming, and thanks to everyone for joining online. So I'm going to talk to you about environmental pollution in the Canadian Arctic, and I'm going to use as an example results from some of the work I did during my PhD. So first I would like to acknowledge my supervisor at the time, Peter Ross, and Lisa Lucero, the Department of Fisheries and Oceans, who was one of the main contributor in that research as well. So first I wanted to put like a few questions up there for you guys to think about and also to take home if you want to do some more research after the course. Some of which I'm going to talk to a little bit tonight. So mostly why is there pollution in the Arctic? Where is it coming from? How does it affect species at the top of the food web, so marine mammals? I'm going to talk about belugas. And are those contaminants a problem for the health of those marine mammals? So first, pollution. So when you think about pollution, you can mostly picture like those fume stacks or end of the pipe pollution. But if you look at the definition, so pollution is the presence in or the introduction into the environment of a substance which has harmful or poisonous effects. So when we want to talk about pollution, where do we start? There are about 100,000 chemicals on the global marketplace, and every year you have about 1,000 new chemicals that are added to the market. So there are a lot of pollutants. So where do we start? Which one are we looking at? So it's a very complex picture. They have a wide variety of physical chemical properties. So some are water soluble, some are going to be fat soluble. And those fat soluble chemicals are going to be the one who are going to have the ability to bioaccumulate in the food chain. So those chemicals are going to be persistent, bioaccumulative, and toxic. So we call them PBT contaminants. So they're going to accumulate in the food chain. They're going to have different ability to induce health effects at the top of the food chain or even in fish species. They also have a range of emission histories, some of them like PCBs or DDT that you might know of. So they've been banned since the 70s. But you can still find them in the marine environment and as well as in marine mammal species, sometimes at concentration that can impact the health of those species. When you look at the marine environment, you have a very complex mixture of contaminants. So some come from local sources, so close to industrial and urban center, you tend to have more contamination, but you also have global sources of contaminants. some of those chemicals have the ability to be transported far away from their point source through the atmosphere or through water currents. So when you think about the Arctic, you think about this remote, pristine environment. And if you look at this map, which show you air pollution measured in terms of particulate matter, so what that shows you, it's particulate matter are a proxy for contaminants. So it gives you a picture of industrial activity in urban center. And you can see that most of the particulate matters are emitted in southern latitudes. So you can see in North America, and then hotspot with those red dots over in Asia. When you look at the Arctic, you can see that there's not much going on in terms of emission. However, many pollutants, as I said before, can travel to the Arctic from southern latitude. The air is one of the most efficient way to transfer pollutants from southern latitudes to the north. It can bring contaminants from the south to the Arctic in a matter of days, so it's really fast transport. And the way it works, so on the graph here on the right, so the contaminants are, we call it grasshopper effect. So depending on the temperature, so the contaminants are going to have the ability to volatilize or being deposited. So they're going to do a series of hops, and then when they're going to reach the Arctic with the cold temperature, they're going to tend to condense and sink in the Arctic environment. On the diagram on the left, so you have once again like the long-range atmospheric transport and deposition into the Arctic. And other means of transport to the Arctic are through river transport or also the ocean currents bringing contaminants to the north. Beluga whales. So that was my study animals for my PhD. So they are long-lived cetacean. They live for about 40 to 70 years. As you can see on this map, they have a wide distribution in the Arctic. And they tend to migrate, so they're gonna occupy different habitats depending on season and depending on where the ice is, and also depending on their food source. So they are typically associated with ice edge in the Arctic. So they're gonna move depending on like the retrieval of the ice during the summer, and then move when the ice is coming back in for the winter. In Canada, there are about 7 different populations of belugas, including a small endangered population in the St. Lawrence estuary. So this population has been classified as endangered. There's about 1,000 individuals left, and they face many stressors, including noise pollution, but also contaminants. As I mentioned before, belugas are ice-associated animals, and those for 2 main reasons. The first one would be the ice, being close to the ice edge, provide them with protection from predators. The belugas have evolved to be able to live in the Arctic where there's ice, so they have no dorsal fin, so they're able to break the ice with their back. And so by being close to the ice edge, they're protected from predators like killer whales. And you have climate change, so you tend to have less ice in the Arctic. So you see in the east, you have the killer whales that tend to like go further north than they used to before. So for them, being close to the ice edge provides some kind of shelter. And the second reason is sea ice is really important for the Arctic food web. Belugas eat a wide variety of species, with their favorite one being Arctic cod, Arctic char. And those fish are highly dependent on the food web that's coming from the ice algae that grow at the bottom of the sea ice. And here you have a picture of the Arctic char. The Western Arctic beluga whale population, so that's the one that we are going to talk about today. So it's one of the biggest population of beluga. There are about 40,000 of them. And every year, in the spring, they migrate from the Bering Sea to the Beaufort Sea, where they spend their summer. So here on this map, here is like their main summer hangout place. So it's the Mackenzie Delta in the Northwest Territories. So they arrive usually in the Mackenzie Delta region end of June, early July. And while most of the individuals are going to stay in this area, some of them, and mainly the large males, are going to continue a little bit further east to spend like late summer in this area here. And when it comes like mid-September, so you have the ice is going to start to form again and push the belugas back west. So they're going to start their migration back to the Bering Sea. When we want to look at the health of belugas, so we need to recognize that many factors can affect the health of those whales. So some of them are mentioned here. So you can have the age is going to influence the health, nutrition, stress, season, disease, and what we're interested in today is contaminants. So many chemicals can be found in the beluga whale food chain, so it's a very complex picture. It's summarized here on this diagram. So in number 1 here you have toxic spill, so that would be an extreme event. Otherwise you can have just contaminants that are being deposited, as I said before, by long-range atmospheric transport, entering the marine environment, and then getting into the food chain. So the belugas can be exposed through inhalation or ingestion, but it's a very minor route of exposure. The main route of exposure is going to be through the diet and through the food chain and bioaccumulation. And once those contaminants are being accumulated in marine mammals like belugas, they're going to have the potential to induce adverse health effects. So when we want to look at the health of marine mammals like belugas, so they give us a good understanding of what's going on in the wild in the population of marine mammals. So it has a high ecological relevance. So you're looking at a particular population of marine mammals. However, it is really important to take into account what's been done in lab animals, because in a lab setting, you can just expose those lab rats to like a specific contaminants to study different pathways and how their toxicity is going to work on a specific organ. So because those animals are going to be exposed, as I said, to a complex mixture. So we need to, when we find something in belugas, we go back to the literature and see what was found in lab animals to support the results that we see in belugas. In the middle here you have seals, so harbour seals. They're kind of in between because there have been a lot of studies done on harbour seals, captive harbour seals or wild harbour seals. And their physiology is very well understood, so it's usually the go-to species when we want to understand what's going on in marine mammals, and also because they're fairly easy to handle and to capture. So here on the West Coast, we have a monitoring program where we use harbour seals to look at contaminant trend and also to look at health effects. So some of the studies, just a few examples of what's been found in terms of contaminant exposure and health effects in marine mammals. So first of all, there have been quite a few studies looking at marine mammals inhabiting industrialized area in the south. So for example, PCBs have been associated with disruption of the immune function, reproduction, and development in seals from the Baltic Sea, California, and closer to here in Puget Sound. Also in captive and wild seals, we've seen disruption of hormone levels, immune function, and enzyme activity are related to PCBs. A couple of years ago, there was a study that was done on the killer whales in our waters here, and it was found that PCBs were altering the gene expression of several genes involved in like reproduction and hormones regulation. And also, I mentioned the St. Lawrence beluga whale population. So it's an endangered population. It's a very small population, and they also have high levels of contaminants. And the PCB levels have been linked to high rates of cancer as well as other disease in this population of belugas. When we talk about the Arctic, which is what we're interested in here, there have been previous studies showing that PCBs have moderate effects on the health of ring seals in Labrador, as well as polar bears. So I did some work with the community of Tuktoyaktuk. So it was a great opportunity to be able to look at Beluga, look at contaminant concentration, and try to figure out if the concentration that we were measuring in those wells were affecting their health. So Tuktoyaktuk, so here you have, it's a little community here on the red star, so it's in the Northwest Territories, and it's about 800 people living in this little community. It's very remote, so here you have a picture in the summer, a picture in the winter, and every summer, so when the whales come in, they go out to harvest the beluga whales. And here is a picture of Hendrickson Island. So it's about like a 45-minute boat ride from Tuk, and it's a key location for beluga harvesting. Because the way it works, the hunters from Tuk go out in the Mackenzie Delta, they hunt the whale, and then they stop at Hendrickson Island, where they butcher the whale, cut the whale to put it back in their boat, and then go back to Tuk. So basically what we did, we camped out on Hendrickson Island. So I was there 2008, 9, and 10 for the whole month of July, and basically we were just waiting for the hunter to come by so we could take samples from the whales. Beluga harvesting is a central part of Inuvialuit culture and practice. I've been there 3 summers, and the weather was really different on the 3 different years, and you can tell when the weather is nice, everyone is going out to hunt. And one year, the weather was really bad, so the hunt was really delayed, and everyone was really anxious to get out because belugas are a very big source of food for those communities. Like it can provide food for months for them, and it's a family-oriented practice. Like you see their hunters, they come out with their family, they teach their kid how to properly butcher the whale, get the maktak, which is the blubber, how to get the meat, and then they go back to Tuk where they prepare. So here you can see like the big pieces of maktak. So that's the blubber, and you can they either eat it like boiled or they dry it. So it's really like a community experience. It's really important for them. It's to keep their tradition going. It's really a teamwork for the collection of beluga samples. So the way it works, so the hunter would come to the island, and they would really help us to collect the samples, and we would help them to butcher the whale as well, so that the process is being done like as fast as possible, and it can go back to the community and process all the meat that they got. So we take a lot of measurements. So here you have an example. So we would take the length, the girth of the whales. We would also get the jaw, so we could get the teeth in order to age the animal. It's important to know the age of the animal when you look at contaminants, because it's known for the persistent organic pollutants like PCBs, for example. Like as you get older, you tend to have higher concentration because it accumulates in your body. And then we take a variety of samples. Here you can see Lisa, she's taking a blubber sample. Here you have a close-up of the blubber. So you have a thick blubber layer and then the skin. So the advantage of working with harvested animals is that you have access to a lot of different samples. As I mentioned before, we also do monitoring here on harbour seals. And we do live capture, so we can only take blubber biopsy and blood. While for these animals, you can take pretty much samples from any tissue you want. So we took blubber, liver, brain, muscle. So it helps you have a full understanding or a better understanding of what's going on in the animal. So in terms of what we found, the Western Arctic belugas are exposed to moderate levels of many contaminants of concern, but you can see on this graph that PCBs and DDTs are dominating the patterns. So here I'm just presenting like total concentration in microgram per kilograms, but really in PCBs we analyzed 209 different PCBs, so this is a total concentration. So just to compare, like I was talking about the St. Lawrence beluga whale, so if we take the PCB concentration here and compare to the St. Lawrence beluga whales, so in the Arctic it's about 10 times lower than what's been measured in the St. Lawrence beluga whales. And if we look at mercury, which is not on this graph, but it's also a major contaminant of concern, the mercury level in the Arctic whales are about 5 times lower than the levels that have been measured in the St. Lawrence belugas. So now that we knew what the concentrations were, we were interested to see if those concentrations had an effect on the health of those whales. So we developed new tools to measure beluga health, and we used molecular techniques to figure out if there was disruption of endocrine system or reproductive system. So basically, here's like a basic diagram of like the full loop. So you have your beluga, you get your sample. So here, for example, it's blubber, but it can be liver. Then we get back down south where we process the sample. So we would extract the RNA, make cDNA, and then we would be able to look at gene expression of different genes of interest. So I'm going to talk about a few genes. And so the first one I'm going to talk about is the aryl hydrocarbon receptor, which is an indicator of dioxin-like toxicity. So PCB compounds are known as dioxin-like compounds because they tend to have similar physical chemical properties, so they tend to behave the same way. So here's like a simplified diagram of what's going on once you have dioxin-like compounds of PCBs entering the animals and entering the cell. So it's going to bind this particular receptor, so the AHR, and then you have this cascade of reaction that's going to happen in the nucleus of the cell. And one of the endpoint is it's going to induce the expression of metabolizing enzyme. So those enzymes are going to be able to try to break down those PCBs. This is what we found in the belugas. So on the x-axis you have PCB concentration, and then on the y-axis you have AHR RNA expression in the liver. And so you have the whales that were sampled in 2008, 9, and 10 with different colors. And you can see that there is a positive relationship between AHR in liver versus PCB. So the higher the PCB concentration, then we found more AHR expression in the whales. You can also notice on this graph that you have kind of this group of whales here, which is the whales from 2009 and 2008, and those whales here, the 2010 whales. So you might ask, why are those down here? Well, it just happened that the whales that were sampled in 2010 were significantly younger than the one that got harvested in 2008 and 2009. And we know through the literature that AHR expression tend to increase with age. So we think that those down here, because they're younger, so they have lower AHR expression. Then we looked at the expression of the metabolizing enzyme CYP1A that I was talking about in my first diagram. So here on the x-axis, once again, you have total PCBs concentration, and then on the y-axis, you have CYP1A expression in the liver. And once again, you see this positive trend. So as you get more PCB concentration, you have more CYP1A expression. So this all makes sense with what's been found in the literature. So going back to my little diagram at the beginning, when I was saying it's important to go back to what's been done in lab studies where you can have good information of pathways to toxicity. So we know that it's been well studied in lab rats. That's what's going on when you're exposed to PCBs. You get increased AHR and CYP1A in the liver. And it makes sense that we see that in this tissue, as the liver is known to be the main organ for like detoxification. So as the animal is exposed to more PCBs, it's going to express more of these AHR and CYP1A, so the body can try to break down those contaminants. So this might be a little complicated, but don't be scared, we're just going to break it down. During this study, so we looked at 13 different genes. So we had to use some kind of multivariate statistical analysis to try to understand what was going on with all those genes and all those whales that we had sampled. So this is a principal component analysis results. So if you look at this panel, so basically you have your different whales. And what a principal component analysis does is you have your 13 genes, and it's gonna pull everything that behave similarly. So you look at patterns. So everything that groups together, it means that they behave like similarly. So on this graph, you have the different whales, and you can see that we have a grouping by year. So you have the 2009, 2008, and then the 2010 whales. And then if you look at the companion panel here, where you have all your different genes, so here are CYP1A and AHR that I just talked about. So we've seen that they behave similarly. So they group together on the PCA, and we know that they are affected by contaminants. We saw that positive relationship with PCBs. And then you can see that you have all these other genes here that are kind of grouping together. So those were the genes that were related to growth, development, and metabolism. And we know that their expression is behaving the same way because they're clustering to that right end of the graph. So when we try to figure out, well, why are those genes clustering together, or why do we get like a yearly separation in terms of like gene expression in those whales, we looked at different factors. So obviously we looked at contaminants, but we also looked at biological factors like age, condition. We also look at diet. We had information, we use stable isotope as a good proxy for diet, so we had that data that can help us understand or give us an idea of if the diet changed during the different years, for example. So if you look along this one, so factor 2 here, you can see that those whales, once again, the 2010 whales, tend to cluster at the bottom, like in the negative side of factor 2. And then you have the 2008 and 2009 that are at the same level along factor 2. And here we found that factor 2 is related to age. So that confirmed what I was explaining before, that age was having an impact on the gene expression. Then if we looked at the other factor, so along this axis, you can see that there's a delineation with the 2008 and 2010 whale tend to be grouped to the positive side of the graph, and then you have the 2009 whales over here. And then factor 1, sorry, was correlated with carbon-13, which is a proxy for diet. So we knew then that age was explaining the grouping along this axis, and then diet was explaining the grouping along this axis. So then we were trying to figure out, well, why would the diet be different in 2008 and 2010 whales? And as I said before, sea ice is very important for the Arctic food web. So then we looked at sea ice extent for those 3 different years. So obviously the general trend for sea ice in the Arctic is a declining trend, but you do have interannual variation. And when we looked at sea ice extent in the area of interest, so the Mackenzie Delta, we found that in 2008 and 2010, we had CIs that were relatively lower than in 2009. So then all those genes had higher expression, and those genes are also related to nutritional stress. So what the conclusion we came to was that those whales sampled in 2008 and 2010 had a slightly different diet because of the CIs was slightly lower than in 2009. And we were able to detect an effect at the molecular level with stress-induced genes related to stress that were upregulated. We also had genes involved in lipid metabolism who were upregulated. Obviously, we only had 3 years of data, so it would be nice to have a longer period to confirm that trend, but we did detect effect at the molecular level. So with those 3 years of study and what's found in the literature, so we have evidence that contaminants present health risks to belugas in the Beaufort Sea. So persistent contaminants and notably PCB, so they were the dominant one amongst the one that we looked at, are affecting beluga physiology. So while the effects that we saw were at the molecular level, so it is hard to figure out what's going to happen at the population level, but what we can say is there's likely an increased risk of reproductive, immune function, and developmental effects. And then another grad student did a study which was really cool on the transplacental transfer of contaminants. Because one thing is, marine mammals accumulate contaminants over time, but for females, one important thing is they're going to offload contaminants to their fetus and through transplacental transport, but also through the milk. So through our 3 year of sampling, we had samples. We had 3 or 4 mother-calf pairs. So we were able to get blubber from the fetus and blubber from the mom. So we would be able to look at the concentration. And Jean-Pierre Desforges is the grad student who looked at that, and he was able to figure out that there's about 12% of the body burden of contaminant from the mom that is being transferred to the fetus. So in terms of PCBs, he looked at PCBs and PBDEs. So which is, it's also another concern from their health, because when you have fetus or newborn, they're more sensitive because they're still developing and having a weak immune system. So being exposed to those contaminants can be an additional concern. So obviously we have some interesting results, and we use some cool new methods to look at health of those marine mammals, but we only had 3 years of data, so we still have lots of more work to do in order to confirm or not the initial results that we found. And that's it. Thank you.
All right, great. So while you folks are thinking of your questions, I had one that I just wanted to make sure that I was following along, because you had some really great graphs there and a lot of data to share, and I was trying to think to myself, how do I kind of distill this down so I can understand it? So you talked quite a bit about the contaminants and the PCBs in particular, and how they affect our RNA or affect the gene expression. So does that ultimately end up in reductions in reproduction, or is that something that just creates stress? Like, what does that mean when their RNA is being affected? What does that look like to the rest of us?
That's a good question. And so when you look at effects at the molecular level, so sometimes it's hard to translate to what's actually happening at the individual level or population level. So in the case of those Arctic beluga whales, which are exposed to moderate level of contaminants. So we haven't documented any problem at the, let's say, the reproductive system or anything. But then if you take the St. Lawrence from the belugas from the St. Lawrence, which are exposed to way higher concentration. So nothing has been done in terms of like gene expression work, but you see mortality and you see reproductive system malfunction. So when you have disruption, for example, one of the genes that I was looking at, also the estrogen receptor, when you see disruption at the molecular level, it can, in the end, impact the reproductive system, but it depends on the concentration, obviously.
All right, great, thanks. And then one other question. So you also showed a slide that talked a bit about sea ice extent, and you mentioned that you only had 3 years of data, but you were essentially concluding that when there's less sea ice, they're changing their diet and they're not producing as many offspring, or they're not producing their stress levels are going up and they're not being quite as, reproductive or healthy. Is that correct?
Yeah, what we found in those years of lower sea ice, so basically we had genes, so heat shock protein, for example, it's a clear indicator of like if your body is in stress, you have increased expression of heat shock protein, and this was one of the genes that was was upregulated, and then you had all those genes that are associated with nutritional stress. So once again, those were based on what's been done in lab studies, where they were stressing out lab rats, like fasting, and you could see you had upregulation of those same genes that we saw in the whales. So they are indicator of nutritional stress as well.
All right, great, thanks. Do we have any other questions from our audience here? Everyone's writing some notes down quickly. We have one question back here. Okay, so just make sure that you repeat the question, please, after you get.
Is there any kind of study looking at the actual population of people that are there that are consuming all of this contaminated meat and blubber?
Yeah, there are. So the question is, are there any studies looking at the actual Inuit population who are eating the belugas and about their health, right? So yes, there are some studies. I didn't work on it specifically, but I've read some papers. And for the community that we worked with, so the community of Tuktoyaktuk, at the end of those 3 years, we had a big community workshop where we presented all our results. And some of my colleagues were there at the same time, and they did speak on it. So I can say some of the things that they say, but I'm not an expert in human health. And for the levels that we see in the belugas, it was still safe for the consumption of like the amount that they consume and the concentration that we see in the belugas. It was fine for this specific community. They did mention that the way, because mercury is also a big concern, and they did mention that the way that they would cook the meat, so for example for the mussel, they would either cook it or just dry it, and the way you would prepare the meat would also affect mercury concentration. So that's something that they have to take into account as well.
All right, great. So do we want to take any more questions before we call it a night? I think I just want to ask our speaker one last, since this is kind of the one ocean pollution effects on the Arctic environment. What is kind of maybe the take-home message that we want folks to remember when it comes to pollution in the Arctic? And there's obviously a connection between what we do here and what happens in the Arctic, because they're not creating PCBs up there. So, is there something that you want people to kind of walk away from tonight, knowing, about pollution up in the Arctic?
Well, I think that what you mentioned is a great message that, like the contaminants that we might release in the environment here, it's not only creating a local problem, like it's really creating a global problem. And as I show, those contaminants can reach the Arctic in a matter of days. So some contaminants are releasing here today, like in 3 days they can be in the Arctic. So it's really important to have that in mind, that the contaminant or the pollution problem is really like a global problem. And the Arctic, everyone thinks, or most people think it might be this pristine environment, but it might not be as pristine as we think.
All right, great. Well, thank you very much, Dr. Marie Noel, for coming and talking with us tonight about ocean pollution in the Arctic. I'd like to remind you that the Vancouver Aquarium Marine Science Center is a nonprofit organization dedicated to the conservation of aquatic life. And if you'd like to make a donation to the aquarium, you can by going to our website at www.vanaqua.org. Thank you very much and have a good night.